Hi, and welcome to our video about the binding kinetics of snRNPs in the spliceosome cycle. This video was made for MCDB 427 Molecular Biology at the University of Michigan. This video goes over figure 14.23 in Robert Weaver's Molecular Biology 5th edition textbook. Before we get to the figure, we will first go over splicing and the spliceosome cycle. Splicing is an important event that happens during processing of pre-mRNA. During splicing, introns are removed by forming a lariat, as seen in step 1 of this figure. Then, the remaining exons are ligated together and further processing can occur. The spliceosome cycle is needed for splicing to happen. The spliceosome cycle takes place with the help of small nuclear ribonucleoproteins, or snRNPs, otherwise known as SNRPs. SNRPs are made of small nuclear RNAs, or snRNAs, that are coupled to proteins. The SNRPs that are active in the spliceosome cycle are named U1, U2, U4, U5, and U6. This cartoon here provides an overview of the spliceosome cycle. Because we are only focusing on binding in this video and not the entire splicing mechanism, we will go over the first part of this diagram only. In this model, U1 binds first to the pre-mRNA. Following U1, U2 binds to the mRNA with the help of ATP. After U1 and U2 bind, U4, U5, and U6 can bind to the pre-mRNA and facilitate the removal of the intron. So I'm sure you're all asking, how did the scientists determine the binding order of these SNRPs? Doctors Ruby and Abelson from Caltech set out to answer this question. To do this, they first attached radiolabeled pre-mRNA to avidin beads by using a biotinylated anchor RNA that was complementary to the 3' end of the pre-mRNA. The pre-mRNA used for this experiment was mutated either in the 3' splice site, which is the mutant C303/305, or the conserved branch point, which is the mutant A257. The reason that they used the splice site mutation was to make sure that splicing didn't go to completion so that they could visualize the binding to the early SNRPs. This occurs because the C303/305 mutant undergoes the first splicing reaction and accumulates spliceosomes. On the other hand, they used the A257 mutant to ask if it perturbed U2 binding. The pre-mRNA was then incubated with nuclear extract at 15 degrees Celsius or 0 degrees Celsius and with or without ATP. From there, they were able to centrifuge to form a pellet to isolate the RNA. The RNA was then denatured, separated by gel electrophoresis on a polyacrylamide gel, and then finally they were able to perform a northern blot with probes to all of the snRNAs. They then were able to expose this to X-ray film to show the labeled pre-mRNAs and snRNAs. So let's move on to figure 14.23a, which is a northern blot. Before we move on to the analysis of this northern blot, we should make a note of the conditions under which everything occurs. As mentioned earlier, some of this experiment takes place at 0 degrees or 15 degrees Celsius and with or without ATP. Also, the scientists used 2 different exposures of this gel in this image, which is why there is an arrow here on the right. To analyze the binding of the SNRPs, first the scientists needed to check the baseline levels of binding. To do this, they loaded lanes with no pre-mRNA and just the anchor RNA. Lanes 14 and 15 ask the question, do any of the snRNAs bind to the bead or the anchor RNA? Because there is no pre-mRNA here, whatever bands can be seen in these lanes are just nonspecific binding to the beads. One can see some bands that are of light intensity for lanes for U1 and U5. Therefore, these need to be accounted for when analyzing the rest of the data. There is no band for the pre-mRNA, which makes sense because no pre-mRNA was added in these lanes. The scientists wanted to use these lanes to determine the binding order of the SNRPs. So now let's move on to the analysis of this gel. For now, I removed lanes 9 through 13 from this image so that it is easier to compare the lanes. Lane 1 is just the marker, so let's look at lanes 2 through 7, which we should note are using the mutant C303/305. This is important so that the experiment stalls and doesn't continue as it would if regular pre-mRNA was used. Going back to lane 2, we can see that there is a darker band at U1 compared to the lanes with no pre-mRNA. This suggests that U1 is binding to the pre-mRNA at 2 minutes. The other bands are found in equal levels as in the no pre-mRNA lane, so they can be considered background levels and can be ignored at 2 minutes. Therefore, Lane 2 suggests that only U1 binds at 2 minutes, and U1 is the first SNRP to bind to the pre-mRNA. In the next lane, the U1 band gets darker, but now you can also see a band at U2. The other bands are still at comparable levels to the background, so they can still be treated as negligible. Therefore, it can be concluded that U2 is binding at 5 minutes, and U2 binds second after U1. Following the same logic, around 20 minutes, you can see that U4, both forms of U5, and U6 start to bind based off of lanes 5 through 7. Lanes 2 through 7 were done in the presence of ATP. Lane 8 shows binding at 60 minutes when there is no ATP present. Comparing lane 8 to 7, because it is important to compare the lanes at the same times, it appears that the level of U1 is about the same, but there is less U2 present. This suggests that U2 requires ATP to bind properly in the spliceosome cycle. So next, the scientists wanted to know what happened to SNRP binding when the temperature was lowered from 15 degrees Celsius to 0 degrees Celsius. The next 3 lanes are done at 0 degrees Celsius. All of the SNRPs are at comparable levels to the nonspecific binding, so it appears that none of the SNRPs bound very well at this low of a temperature, with or without ATP. Lanes 12 and 13 are done with the A257 mutant. This is the branch site A mutant, where a uracil replaces the branch site adenosine, reducing splicing by around 95%. From the low levels detected, we can see U2 does not bind with this mutant. U1 has a light band that appears to be close to the background amount, so this looks like it indicates that it is not binding. Looking down at the other SNRPs, we see that they too are not binding to this mutant. However, it also appears that the scientists loaded less pre-mRNA in these lanes, as seen by the fainter pre-mRNA bands. The pre-mRNA bands in lanes 12 and 13 look much fainter than those in the other lanes, such as lanes 9 through 11, so we should consider this in our conclusions about this data, especially for U1 binding. There's a lot that we can conclude from this figure. The first thing that we can conclude from this figure is that U1 binds first to the pre-mRNA. We know this because the U1 band appears first on the gel after we account for nonspecific binding. Following the same logic, we can conclude that U2 binds after U1. Another thing that we can conclude using the same logic as with U1 and U2 is that U4, U5, and U6 bind after U2 and U1 bind. A fourth thing that we can conclude is that U1 doesn't require ATP to bind, but U2 does. We can conclude this because when no ATP is added, there is a reduced amount of U2 in lane 8, but the amount of U1 remains about the same. Another conclusion that we can make from this data is that U1 binds only at 15 degrees Celsius and not 0 degrees Celsius. Finally, we can conclude that there is no U2 binding with the branch point mutation. Keep in mind that it appears that the scientists did not load equal amounts of pre-mRNA in these experiments, so it is hard to make definite conclusions about the relationship between U1 and the branch mutation. Before we end this video, let's go back and make sure that this data matches the diagram that we introduced at the beginning of this video. Like we mentioned, U1 binds first, as seen at the top of this image. Then U2 can bind with the help of ATP. Finally, U4, U5, and U6 combine to help complete the splicing event. We hope that this video is helpful in understanding the order of the binding of the SNRPs that occur in the spliceosome cycle. Thank you so much for watching, and remember, go blue!